My name is Mark Smith, and I'm currently a consultant for American Preclinical Services, APS, a Minneapolis contract laboratory focused on testing medical devices. APS was recently acquired by NAMSA, one of the major global players in med device testing, and we continue to play a major role in medical device testing and consulting with a great deal of experience in neurotechnology. I've been in the business of pharma and medical device discovery and development for over 30 years. I also participate in a few of the ISO committees as a voting member and have participated in some of the sausage-making committee meetings wherein these standards were annually discussed, refined, and updated. Let's review the learning objectives for this session. Novel medical device systems require demonstration of safety through the following: biocompatibility evaluation following the International Standards Organization or ISO 10993 standards, sterility assessment standards, and other standardized testing including hermeticity, electrical safety, and MRI safety, all of which also have their own standards. We'll focus today on the ISO 10993 standards and look at some examples to understand how the characteristics of the device and components determine the required testing plan to meet these standards. We'll discover that complex devices require the creation and optimization of well-planned testing schemes along with careful allocation of resources. Through examples, we'll review device characterization, sample preparation, extractions, device allocation, selection of required testing panels, and finally some hints on interpretation of unexpected results. Let's define and identify the driving standards required for testing these complex devices in the area of biocompatibility. How is biocompatibility defined? What are the ISO 10993 standards? And how are the ISO standards interpreted by the FDA to assure device approval? Biocompatibility is a characteristic of a device or device component denoted by its compatibility with biological systems, as indicated by an appropriate or acceptable host response of the biological system to the deployed finished device. In general, the factors influencing biocompatibility include the following: the material from which the device is manufactured, the manufacturing process, including any additives, lubricants, or release agents, which may be integral parts of the build, surface properties such as specific geometry or coatings, interactions between these materials of composition, and finally changes in the device due to the effects of the biological system or tissue, such as biodegradation, or any subsequent chemical change of the device over the duration of its implantation or use. Often manufacturers will assume that because their device is composed of materials that have historically been proven safe, they will require minimal testing. However, although the materials themselves may be safe, until we combine the potential impact of any chemical additives, coatings, unanticipated interactions among the materials, and potential degradation, in combination with any unexpected effects that the physical surface of the finished product may have on the biological system, we cannot claim safety according to these standards. Regulatory agencies also take this approach and will rarely allow a new product made of old and common materials to proceed unless the full complement of testing standards are met. How do we approach satisfying this wide range of required testing? The good news is we have guidelines. ISO 10993 standards guide the biological evaluation of medical devices over a risk management approach. This guideline contains over 20 parts. Part 1 is a guidance to overall risk management. Part 2 describes the overarching animal welfare requirements of the testing processes. The remaining parts will focus on the individual endpoints and also provide specific details as to how to perform the tests. Before we see how to apply these standards, let's look at some definitions associated with the wide range of differing medical devices, even if we stay focused in the area of neurotechnology. When we talk about medical device biological system contact, there are 4 basic types. The first is non-contact, which should be obvious and does not require any biological testing. This category is limited to items such as software or materials contained completely inside impervious coatings or coverings with no potential for leaching to the patient. Transient contact specifies that the device is either transient or in passing with skin or other tissues. This describes many devices such as the lancets, needles of interaction less than 1 minute. Direct contact occurs when the device comes into physical contact with skin or body tissues. Indirect contact, on the other hand, means the device components do not have physical contact with the body, but a fluid or gas can pass through or around the device prior to reaching the patient. For example, an IV bag or an airway tube where the internal lumen of the device will not have physical contact with the patient, but gas or liquid inside the bag can pick up any leachable compound from the lumen, which could then be delivered to the patient. In this case, the risk is still there even without physical contact. Slide 9. When we classify devices in order to design the testing panel for biological evaluation, we will primarily focus on the direct and indirect contact portions, and we will further characterize the devices by the nature of the contact. An example of surface contact would be a surface probe or monitor for a transmitter. External communicating devices would function as a catheter in the cardiovascular area or a neurotransmitter with externalized leads. Finally, we'll have implantable devices such as coronary stents, spinal stimulating leads, or deep brain stimulating electrodes. Devices are also characterized by the duration of contact. These categories are straightforward depending upon the nature of use, as limited, prolonged, or when greater than 30 days, they're denoted as permanent. Now we get to the heart of the testing standards. Table A1 is a guide that summarizes the expected testing endpoints based upon the category of the device or device component and the nature of its contact. As you can see from the density of the X's, the more invasive the contact and the longer the duration of use, the more testing is required. Let's dissect this table a bit more and evaluate the breakdown in device categories. We can have surface contacting devices. These are direct contacting devices but are limited to external applications such as intact skin all the way through to breached or compromised skin or mucosal membranes. Next, we have the full range of external communicating devices such as electrode leads with external access, bone pins, catheters or electrodes, and intravenous devices with a full contact with circulating blood. Finally, the most complex devices, which are destined for spinal or brain implantation with either tissue or blood contact. Now let's look at the range of biological endpoints. Beginning with the essential physical and chemical characterization of the device materials through a whole series of biological assessments. These assays can be as simple as evaluating device extracts for cytotoxicity over 72 hours with mammalian cells in culture, all the way to animal testing as extensive and rigorous as carcinogenicity or reproductive toxicity testing. Again, each part in the list of ISO 10993 standards carefully defines the nature and scope of the testing required as defined by each of these columns. This table was updated in 2018, and there was one new column added that is highlighted here. This is a newly emphasized requirement for chemical and physical information. This information can be sometimes obtained from your material vendor, or it can be obtained by direct and expensive chemical analysis. But remember that chemical information is also informed by some of the other steps in the complex manufacturing processes, including additives, ink, mold release agents, and such. Overall, this table is memorized by all professionals from biocompatibility testing laboratories and is an extremely useful tool for meeting the standards and satisfying the FDA. In September of 2020, the FDA issued a new document which provides their interpretation for ISO 10993 Part 1. This document supersedes the earlier versions that the FDA had published in 2016, and this guidance document, in combination with Part 1 of the standards, will direct us in designing the correct panel of tests for the overall biological biocompatibility assessment. Representatives from the FDA had a significant input in the development of the ISO standard table A1. but they also constructed a similar but not exact copy in their guidance document, one which is not necessarily in exact agreement with the ISO committee interpretations, but which rules in all discussions and submissions for device approval to the FDA. The differences are minimal, but are reflected in this table in the newest version of their guidance document. First, although there is no chemical characterization stipulated in the FDA version of the table seen here, there continues to be strong requirement for this information to be included in FDA submissions and risk assessment plans. In addition, the FDA document differs from Table A1 in a few other ways. As an example, for a surface contacting device interacting with mucosal surface, there is no requirement for materials-mediated pyrogenicity testing in the ISO standards. However, the FDA has recommended this test still be performed. In many instances where a company has developed and performed their biological evaluation plan strictly according to the ISO 10993 standard, they will be asked to add this test if it's submitted to the FDA. Now let's look at some examples from the area of focus for this program, Neurotechnology Devices. We'll discuss surface contacting components such as controllers or data acquisition systems. Additionally, we could consider implantable leads or electrodes with hardwired external communication. Finally, we'll look at fully implantable devices with external signal collection or energy delivery management components. There can also be a wide range of duration of use across these different systems, from short-term to permanent implants. Let's review some hypothetical devices and device systems in terms of their contact and duration. So here's a headband with an external surface contact and separate non-contacting controller with an external leads. This system would likely have short to long-term contact duration. The second, more complex brain-stimulating system may have fully implantable tissue and blood contacting device components. However, it may be fitted with a non-contacting data controller. This configuration would likely be for long-term use. Here's a GI device with a neurostim function, again with an implantable electrode and implantable pulse generator IPG. There would be another completely external component for controlling device performance and data collection. Again, this system would have prolonged to long-term contact duration. And the final, more complex brain-stimulating system may have fully implantable tissue and blood contacting components with a connected surface communicating interface module and a wearable external contacting controller device, again likely for prolonged to long-term contact duration. Let's dissect the final example of a deep brain stimulating device according to Table A1 and identify the required testing. Start with the stim electrodes that are permanent implants with deployment directly into intact tissue and with leads following a path through bone and subcutaneous tissue. Follow the categories in the table, and we can see that these components would require an extensive panel of testing. Next, we isolate the cable, which can be tested as a separate component with external communicating characteristics. Again, identify the boxes recommended for testing. In this case, these two components are similar in their testing requirements and may therefore be tested together, potentially saving significant time and resources. The external cable, harness, and controller require a less extensive list of tests and would therefore be submitted for testing separately. Also, as this testing is not as extensive, there may be material required in this component that may not need the rigorous testing of the other two, thereby allowing for different materials or processes to be employed in the manufacturing. For example, colorants could be used if desired, or other inks or markings applied, and even with these extra materials of composition, this component of the device could be subjected to a less rigorous testing panel. Finally, the internal wires and components of the stim electrode, the internal workings of the IPG, and the cable are all non-contacting, and therefore they do not require any separate testing. This is key as long as they remain sealed. There is much more freedom in selection of these materials while designing the manufacturing process. Remember, this is a guidance, especially when submitting to the FDA, and many tests can be combined. These combinations would require justification on a case-by-case basis, ideally with discussions with the target regulatory group. However, this guidance is very valuable in beginning the design of the Biological Evaluation Plan, also known as a BEP. What we just did are the first steps in constructing a BEP. You can do it yourself. However, you may also ask your friendly contract lab for some help. It's sometimes free when you agree to perform the testing with that lab, or if it requires extensive planning and complex risk assessment, it can be obtained at a nominal cost. Overall, it's a guide for the next stages of meeting these compatibility standards. First component of the BAP is a detailed device component breakdown, including material composition, additives or colorants, and processing steps. These are followed with a careful component-by-component analysis of contact and maximum duration of use. We see here the actual biological endpoints corresponding to the ticked boxes which we identified from the recommendations in Table A1. This table shows the big 5 biocompatibility assays most commonly run and required for nearly all minimally invasive devices, along with the accepted extraction methods, assay conditions, and target results, which, if attained, will qualify the device as safe with respect to these specific endpoints and these particular standards. This continuation panel lists the more complex, rigorous, and longer duration biological assays, which are also frequently utilized for well-characterized devices to assure compliance with this set of standards. The longer duration and implantable components of our device would all require these tests be performed as well. Let's digress for a moment and look at exactly how one goes from a device in final finished form in its packaging to testing in a biological assay per these standards. First, except in the case of implantation tests, remember that we're not actually testing the intact devices, but evaluating the performance of an extract of each device in both a polar and a non-polar solvent, typically normal saline or sesame seed oil, in these bioassays. These extracts would be assumed to contain any leachable or extractable chemicals from the device components that could interact with the biological systems. These animal models are then exposed to the extracts by appropriate routes and dosing, and the results read out after the appropriate duration, from overnight up to 26 weeks. Remember that planning your timeline is a key to implementation of a successful biological testing plan, especially when filing deadlines are present. The number of devices required for testing needs to be calculated to assure that sufficient material is available. Some programs require testing of hundreds of devices at considerable expense to the manufacturer. These need to be final, finished devices, ideally in final packaging. Also, the suitable subcomponents of each device must be identified and submitted for appropriate testing in each panel. Proper controls may be required depending upon any exceptional characteristics of the device. And finally, there may be an option for use of a coupon or representative sample made from the same materials and subjected to identical manufacturing steps. These coupons are especially useful when the size and shape of the device is not amenable to actual testing, such as in the case of an implantation study. It's difficult to put a full-size human heart valve into a rabbit, whereas a coupon would be suitable for biocompatibility testing for implantation, as well as for most of the other tests, and can save considerable time and expense versus utilizing the final product. Now that we have the extracts prepared and the test completed, let's look at the results, which most often are pass or safe and require no further work and lead to a successful submission. However, when a biocompatibility test fails, we need to marshal resources to address the issue, find the cause, and minimize the impact. Did anything go wrong with the tests, including study deviations, unexpected animal variability, or contamination? Was the failure clinically significant in the manufacturer's eyes versus the eyes of the FDA reviewers? How have the interactions with the individual reviewers and the regulatory teams, the FDA, been throughout the submission? And might there be issues in communication which led to an overinterpretation of the clinical impact of a failed result? Troubleshooting is the main job of our scientists in the Biocompatibility Lab. We try to identify hidden sources of test failure along with the clients. Some things should have been expected, but testing failures always require careful evaluation of what happened and why. Sometimes surprises occur, but we should have known. Glutaraldehyde is often used to sterilize and preserve biological materials in devices. However, no matter how well a device is rinsed, per stipulations in the IFU or instructions for use, extracts from glutaraldehyde-treated materials will always fail a cytotoxicity test. These failed tests need to be justified, and this is a good example that most always meets with the reviewer's approval. Skilled managers run testing labs, and trained technicians perform the tests, but these are people, and people occasionally make mistakes. The good thing is that all these assays are performed under good laboratory practices, GLP, and everything done in these GLP assays should be well documented, so it's not too hard to find mistakes and either justify the results or justify rerunning the tests at the expense of the lab, not the manufacturer, and not lay blame on a true device safety issue. Also, were the correct subcomponents tested appropriately? Many devices, as the examples we reviewed earlier, are composed of many separate parts, and the testing may only use selected components for a specified test. The correct components must be selected as the guidance we reviewed earlier stipulates. Also, when devices are processed for extraction, it's common for the device to be cut into pieces. Occasionally, internal components such as wires, circuits, or connectors, which should be shielded by the construction of the system or by special coatings, may accidentally become exposed to the conditions of the extraction solvent and could lead to test failure. This is one example of improper extraction. The volume of the solvents used in the extraction is dependent upon surface area of the device, and mistakes in calculation of surface area are a common source of improper extractions, which may also lead to repeated tests. Occasionally, the failures can be attributed to the manufacturer. For example, a wrong batch was slipped into the testing stream, or a material may not have been exactly as represented by a vendor. Occasionally, a great place for discussing failed test results is in the area of clinical significance. Conditions specific for the device and its utilization can sometimes allow a failed test to not delay a submission. Again, this requires a good regulatory team at the testing laboratory and a lot of experience in dealing with regulators. We'll briefly describe some of the many other standards required for any implantable or direct contacting device, specifically sterility. There are methods of sterilization which need to be compatible with the materials of construction and suitable guidelines for confirming the sterility of the devices in final finished form. Sterilization Category A, which includes methods well established with FDA-recognized consensus, such as dry heat, ethylene oxide in a rigid chamber, steam, radiation, or other methods, all of which have their own unique complement of standards as shown here. Devices requiring sterilization methods in Category B may need more of a case-by-case evaluation and justification, as there are no FDA-recognized consensus standards, but there is published evidence on effectiveness. Examples of these processes include hydrogen peroxide, ozone, and ethylene oxide exposure in a non-rigid chamber. Finally, there are novel methods with little to no published evidence on effectiveness. Examples include high-intensity light, vaporized peracetic acid. These also require special care and justification for use and good communication with the regulatory body to assure that the qualifications and claims for sterility in the submission package are met. Small batch sterilization is another common approach to sterility assessment. It's especially favorable for cases from all therapeutic areas, including neurotechnology devices, which at early stages in development are limited in quantity. That is, the device is too expensive to produce or still in development as part of an IDE submission on a new device. Batch sterilization with ethylene oxide can follow ISO 11135 cases, where the batch is exposed to half cycle followed by full sterilization cycle and tested for functionality and ethylene oxide residuals. All in a customized process requiring appropriate justification and discussions with the regulatory group. This will satisfy the requirement for sterilization without meeting the rigorous requirements for a full sterility validation package. There are a few additional standards to be aware of, including hermeticity testing. Implantable neurotechnologies often require hermetic packaging to prevent moisture accumulation. There are methods available such as vapor analysis using helium, krypton-85, and other noble gases to confirm hermeticity. Environmental testing, such as the performance of the device after exposure to temperature and pressure extremes or mechanical changes, also monitor under an appropriate ISO guidance. Battery testing to ensure operation that avoids overheating and short-circuiting also come in with their applicable standards. Electromagnetic compatibility, or EMC, is yet another area for qualification, including both electromagnetic interference testing and electrostatic discharge testing. Devices for these assessments can be separated into non-implantables and active implantables, again, each with their own applicable standards. And finally, MRI compatibility and safety also have their own specific standards evaluating both patient safety as well as maintenance of device functionality in the presence of high magnetic fields. Hopefully, we covered biocompatibility and the overarching ISO 10993 standards, including how I characterize my device, and how do I select my testing panels, and where can I get help interpreting results that are unexpected. And finally, what other standards should I be aware of? Here are a few other resources that should prove useful in continuing your journey through this sometimes frustrating but remarkably important segment of medical device testing and development. Thank you for your attention.